Hello students, today we are going to discuss a very good topic of pharmacological and toxicological screening methods to as per PCI syllabus for PG students, and this topic is also helpful for those which are concerned with the biomedical sciences. So chromosomal aberration, we will going to see the definition, types and example of chromosomal aberration. So first, we will see the definition. Now, chromosomal aberrations or chromosomal abnormalities. It occurs when there is a defect in the number of chromosomes in a cell of an organism or the arrangement of genetic material, that is genes, on the chromosome. So these abnormalities which is related to chromosome is occurs when there is a defect in these two parts, that is the number of chromosomes in a cell are altered or the arrangement of genetic material, that is genes, on the chromosome is altered. Now, chromosomal abnormality, it gives rise to specific physical symptoms. However, the severity of this depend on the type of aberration. So, alteration or chromosomal abnormalities will lead to some physical symptoms, lead to some disorders, and it depends on the types of alteration which is occur, the type of abnormalities occurs on the chromosome. Now, these abnormalities on the chromosome is maybe in the form of additional material attached to a chromosome rather than the normal material. A whole chromosome is missing, or even in the faulty formation of the chromosome. So, this abnormality you can observe in these three forms. There is additional material attached to the chromosome. Sometimes the whole chromosome is missing, or the faulty formation of chromosome can occur. So these are the abnormalities. Now the types of chromosomal aberrations. These are, if you observe here, these are divided into mainly two types. First is the numerical and second is the structural. The numerical is further divided into euploidy and aneuploidy. Under euploidy, triploid, tetraploid and polyploid, and aneuploidy, monosomy, trisomy and tetrasomy. In structural aberrations, you can observe the deletion, duplication, inversion and translocation. Deletion may be terminal or intercalary. Duplication may be terminal or intercalary. Inversion may be pericentric or paracentric, and translocation may be reciprocal, non-reciprocal or Robertsonic. So, how will you identify the chromosomal aberration? For that purpose, it is suggested that there is a term which is called as karyotype. It refers to the set of chromosome from an individual. So, from an individual, a set of full chromosome is called as the karyotypes. So, abnormality can be identified by detecting or confirming the karyotype comparison of a given genome to a normal karyotype or a species through genetic testing. So, you can do genetic testing and there you compare the karyotype of this abnormal to that of the normal, from which one can come to know the chromosomal aberration. So, chromosomal abnormalities usually occurs when there is an error in cell division following meiosis or mitosis. So, this aberration is usually occurs during the process of cell division, that is meiosis or mitosis. Then first we will see the numerical aberration. So here you can observe it is the ploidy, that is euploidy and aneuploidy. Under euploidy, monoploidy, diploidy, and under that again it is further classified. So as it is a numerical aberration, so here the quantitative terms is very much important. And another is the aneuploidy, where there is a monosomy and nullisomy. So we will see in detail what is this terminologies are there. So actually the number of chromosomal number varies or variation occurs or involves the either the addition or loss of one or more chromosome, which is called as the aneuploidy, and addition or loss of one or more haploid set of chromosome, that is called as the euploidy. So there is a difference between the euploidy and the aneuploidy. So aneuploidy is nothing but addition or loss of one or more chromosome from the chromosome, and euploidy is the addition or loss of one or more haploid set of chromosome, that is the euploidy. So let's see the detail. First we will go with the aneuploidy. When an organism gain or loses one or more chromosome but not complete set. Here it is very important to understand not complete set. This condition is called as the aneuploidy. Only the organism loses one or more chromosome, but the complete set is not loses. It is called as the aneuploidy. Aneuploidy leads to the variation in the number of chromosome but not involve the whole set of chromosome, as this is given. The nuclei of aneuploidy contain chromosome whose number is not a true multiple of basic number. So actually the true actual number of the chromosome you will not find in the cell nucleus of the person if there is a variation in the chromosome. So let's see some of the example where this aneuploidy is occurs. So first is the Down syndrome. So here if you observe it in Down syndrome, the person nuclei having 47 chromosome instead of 46, means one chromosome is added into the set. Another example is Turner syndrome, where 45 chromosomes observed instead of 46, means one chromosome is less. So ideally 46 is required. Here one is added, here one is deleted or removed. So the number of chromosome in the individual is not a true in this aneuploidy as multiple of the basic number of n. So basically it is 23 into 2, that is 46, which is not observed in the Down syndrome and also Turner syndrome. So it is called as aneuploidy. Then what are the types of this aneuploidy? First is the monosomy. Now monosomy is the loss of one chromosome produces a monosomy 2n minus 1, and the condition is known as monosomy. The example which we have seen is the Turner syndrome, where the number of chromosome is 45. Another is the trisomy. The gain of one extra chromosome produces trisomy, and the condition is called as the trisomy. Example we have seen, that is Down syndrome, where one chromosome is more, that is 2n plus 1 is equal to 47 chromosome in human. Here trisomy 18 it produces Edwards syndrome, where additional copy of chromosome 18. Trisomy 13 it is Part2 syndrome, have an additional copy of chromosome 13, and trisomy 8 Varkini syndrome 2 have an additional copy of chromosome 8. So here in everywhere you will find the increase in the chromosome number, that is the trisomy. Then next is the tetrasomy. Here there is a 2 extra chromosome produces, which is called as the tetrasomy 2n plus 2, and the condition is called as the tetrasomy. Example, that is XXY syndrome, then tetra X syndrome, which containing 48 chromosomes, and this syndrome which containing the 48 chromosome. So if you are find here, the chromosome number will increases with the number of 2. Then another class is pentasomy. Here the 3 extra chromosome produces pentasomic, means 2n plus 3, and the condition is called as the pentasomy. Example is penta X syndrome, where you can observe the 49 chromosome. Ideally, it is 46. Addition of 3, it produces 49 chromosome. Female have 5X chromosome instead of normal 2. Signs are intellectual disability, short height, less muscle tone, and delayed in the development of the individual. Then next is the newlysomy. It is the condition in which a pair of homologous chromosome is completely lost. Human with this disorder will not survive. This is very important. So about 21 newlysomic of the allohexaploid triticum aestivium has been made. They differ in appearance from normal hexaploids and have less vigor. So these are the types of this numerical. Then you can observe this is given the types of aberration, aneuploid, monosome, all these things which we have discussed right now, they have given in this tabular form. It will be easy to remember for you. Then another is the euploidy. So what is this euploidy? When one or more complete haploid set of chromosomes are involved in the aberration or involved in the abnormality, the resulting abnormality is called as the euploid. So here there is a complete set of chromosomes is involved. In the previous we have seen only one chromosome is involved. So here haploid set of chromosomes are involved, and that is called as the euploidy. It is more tolerated in plants rather than the animals. And for example, if there is a human cell that has an extra set of 23 chromosome, it will have the euploidy. What are the types of euploidy? Ploidy refers to the number of homologous sets of chromosome in the genome of the cell or an organism. Each set is described by the small letter, that is N. It is very important to understand. Now monoploidy. What is monoploidy? The state of having a single set of chromosome is called as the monoploidy, and it is represented by 1N. It is monoploidy. Monoploidy is lethal in animal, but in case of plant species, this can be more tolerated. In most animal species, this could mean death, but there are few animal species where monoploidy is a normal part of the life cycle, such as male wasps, ants, and bees. So in this case, you can observe this monoploidy. The offsprings that have arisen from monoploidy are those that have developed from unfertilized egg. Then polyploidy, the condition in which a normally diploid cells or organism acquire one or more additional sets of chromosome is called as the polyploidy. In other words, the polyploid cells or organism has 3 or more times the number of haploid chromosome. Then polyploidy arises as a result of the total non-disjunctional of the chromosomes during mitosis and meiosis. These are the reasons why polyploidy happens. Then this polyploidy is mainly observed in the plants. So polyploidy is common amongst the plant and has in fact been an important source of speciation in angiosperms, particularly important in allopolyploidy, which involves the duplication of chromosome in a hybrid plant. A diploid hybrid is sterile because it does not have the homologous chromosome pairs necessary for successful gamete formation during the meiosis. In the case of tetra polyploidy, so it is a diploid hybrid, it is a tetra polyploidy. The plant duplicates the set of chromosome inherited from each parents. Meiosis can occur because each chromosome will have a homologous derivative from its duplicate set. And the tetra polyploidy, it confers fertility on the previously sterile hybrid, which therefore attains the status of a complete species distinct from either parent, and up to half of the known angiosperm species have been estimated to have arisen through polyploidy, including some of the species most prized by men. and the plant breeders use this process treating desirable hybrids with chemicals such as colchicine, which are known to induce polyploidy. So this is the polyploidy which happens or observed in the plant. Polyploid animals are much less common and the process appears to have had little effect on the animal speciation. So polyploidy mainly you can observe into the plant. Then second, that is the structural aberration. Up to now we have seen now numerical aberration, now structural aberration. So these occurs due to loss of genetic material or the reorganization in the location of the genetic material. So two main factors are there. Either there is a loss of genetic material or there is a reorganization in the location of the genetic material, which called the structural aberration. So here it includes deletion, duplication, inversion, ring formation and translocation in the chromosome. Unbalanced rearrangement include deletion, duplication or insertion of a chromosomal segment, or balanced rearrangement induce inversion or translocation chromosomal region. So if you observe here the structural aberration, you can observe the deletion, the duplication, inversion, substitution and translocation. These are the different process, different types of structural aberration. So we'll see the type of structural aberration. First is the deletion. What is meant by deletion? A part of chromosome is missing or removed. Known disorders include Wolff-Hirschhorn syndrome, which is caused by partial removal of the short arm from chromosome 4. So a partial removal of the short arm of chromosome 4 is occurs. And another syndrome, that is Jacobsen syndrome. It is also called as 11q terminal deletion disorder. It is caused due to the removal of the q arm of the chromosome 11. It is the deletion. These are the examples. Now what is duplication? A part of chromosome is duplicated, resulting in additional genetic material. Known disorders include Queteau-Marie-Tooth disease type 1A, which can be caused by duplication of the gene encoding peripheral myelin protein 22 on chromosome 17. Third is the translocation. When a part of chromosome is transferred to another chromosome, there are two main types of translocation. One is the reciprocal translocation, where the segments of two different chromosomes have been exchanged, and another is the Robertson translocation. On one complete chromosome has joined another in the centromere. These only occurs with chromosome 13, 14, 15, 21 and 22. Then next is the inversion. A part of chromosome has been broken upside down and reattached. Therefore, the genetic material is inverted. Then insertion. A portion of one chromosome has been deleted from its normal place and inserted into another chromosome. It is called as the insertion. Then isochromosome. An isochromosome can form when one arm of the chromosome is missing and the remaining arm is duplicated. And last is the rings. Ring chromosomes can result when one chromosome undergo two breaks and the broken ends are fused into a circular chromosome to form a ring. This can happen with or without loss of the genetic material. So these are the various types of structural aberration. And last, how chromosomal aberration or abnormality occurs. So chromosomal abnormalities can occur as an accident when the egg or sperm forms or during the early stages of fetal development. The mother's age and certain environmental factor may play a role in the occurrence of genetic errors. Most chromosomal abnormalities occurs as an accident in the egg or sperm and are therefore not inherited. The abnormalities is present in all cells of the body. However, some abnormalities can occur after conception, result in mosaicism, where some cells have the abnormalities and other do not. And prenatal exams and tests can be done to examine the fetus chromosomes and detect some but not all type of chromosomal aberration. So this is all about the chromosomal aberration. Thanks for listening the topic. If you like this, if you like or if you need such types of more presentation, so kindly like, share, subscribe and give your positive comments on the topic. Thank you.